Lesson number 9, Genomics. By the end of this lesson, you should know how to describe genomics. DNA consists of thousands of building blocks called nucleotides. Each nucleotide has a base as part of its structure. There are 4 different bases: adenine A, thymine T, guanine G, and cytosine C. A gene is a section of DNA. The differences between one molecule of DNA and another, and therefore one gene and another, are the result of a difference in the order of bases along it. The order of the bases of our genes is our genetic code. The code is a set of instructions for combining amino acid units in the correct order to make a molecule of protein or part of a protein. The instructions needed to arrange an amino acid unit in its correct place in a protein molecule are contained in a row of 3 bases, for example GCC, ATG, GAT, CAA. The order of the groups of these bases control the order in which amino acid units are combined, producing a particular protein. A genome is all of the DNA in each cell of an organism. Genomics is the science of working out the sequence of the bases in the strands of DNA which make up a genome. In 1986, scientists in the UK and the USA set up the Human Genome Project to sequence human DNA. The scientists have now worked out the order of the bases of 3.1 billion nucleotides which make up the DNA of a human genome. How did we read the letter sequence of the human genome? Samples of cells were taken from anonymous volunteers. The chromosomes of the cells were broken up into pieces to get at their DNA. Thousands of copies of the pieces of DNA were made to provide enough material to work on. The DNA was then placed inside machines called sequencers, and the most probable order of bases displayed on a computer screen. A gene is identified by comparing the sequence of its bases with the sequence of bases in known genes. However, the question is, which genes code for which proteins? The new and developing science of bioinformatics is helping us to answer this question. Its methods involve the use of powerful computers to help match the base sequences of genes with the proteins they code for. The aim is to find which genes code for which proteins, show in which cells a gene is active, and show when each gene is active. Bioinformatics is helping to make a molecular atlas of the cell. The map will show where the proteins coded for by our genes are normally found.